Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Think about this for a second. Globally, more than 1 in 10 people will face major depression sometime in their life. That's huge.
It really is a staggering number.
And we all talk about stress, the pressure of daily life. But what's actually happening inside our brains under that kind of strain? How does that connect to something like depression?
Well, that's exactly the biological puzzle we're getting into today. We've been digging into this fascinating scientific paper. It looks at how stress impacts a specific brain area, the lateral habenula.
Lateral habenula.
And then critically, it focuses on a cellular process called autophagy within that region, basically cell cleanup.
Yeah.
And how disruption there might, open the gate to depression.
Okay, lateral habenula and autophagy. Let's unpack those a bit. The lateral habenula. My understanding from the paper is it's like a stress response center. Gets really active when we're stressed.
That's a good way to put it, yeah. It's definitely highly responsive to stressors. And this specific study, it's titled, Stress Dynamically Modulates Neuronal Autophagy to Gate Depression Onset. Quite a mouthful, but it really drills down into that cellular level.
How did they study it?
They used various experimental models, mostly in mice, employing some pretty sophisticated techniques to see what's going on. And we'll also bring in some insights from, health news about how antidepressants might fit into this picture.
Got it. So our mission today is basically to connect the dots. Stress, this autophagy cleanup process in the lateral habenula, and how that relates to depression onset. We want to make sense of why this research feels so important.
Exactly. A shortcut through the science.
All right, so let's jump into the core findings. They found something key about how different kinds of stress affect this autophagy process, didn't they?
Yes, and this is really interesting. It's almost like a two-sided coin. They found that short-term stress, what they call acute stress, actually activates autophagy in the lateral habenula.
Activates it, like turns it on.
Yeah, cranks it up. You can sort of picture it like the brain sending in a rapid cleanup crew to deal with a temporary mess.
Okay, that makes sense. A quick response to restore order. But what about long-term chronic stress? That was different.
Completely different. Under chronic stress, autophagy in that same area gets suppressed. It's dialed down.
So the cleanup crew gets overwhelmed.
That's a great analogy. They start strong, but under constant pressure, they just can't keep up, and the whole process slows down. So it's not that stress is always bad for autophagy. Short bursts seem to kickstart it, maybe protectively.
And they saw this consistently. The paper mentioned different stress models.
They did, and that really strengthens the finding. They used restraint stress, social defeat stress, which is like intense social pressure for mice, and even foot shock stress. Across all these prolonged stressors, the pattern was the same. Less autophagy in the lateral habenula.
And it wasn't just about feeling stressed,
Yeah.
right? They linked it directly to brain cell activity.
That's right. They actually managed to mimic the neuronal firing pattern seen during acute stress, specifically where the lateral hypothalamus connects to the lateral habenula.
So they artificially stimulated that connection.
Exactly. And even that artificial simulation was enough to boost autophagy in the lateral habenula. It strongly suggests the activity of these neurons during short stress is what triggers the cleanup.
How did they actually see these changes? You mentioned looking at the cells. Something about LC3 puncta.
Precisely. They needed visual confirmation. LC3 puncta are like tiny fluorescent flags that light up where autophagy is happening inside the cell. More flags after acute stress, fewer after chronic stress.
Yep.
And they went deeper with electron microscopy too. That gives you a super detailed view. They literally counted more autophagosomes, the little sacs that engulf the cellular junk after acute stress, and fewer after chronic stress. Direct visual proof.
So stress flips this autophagy switch.
Okay.
Now, how do antidepressants fit in? The study looked at that too.
Right, and this is where it gets really relevant for potential treatments. There are already hints that autophagy might be involved in how antidepressants work, so these researchers specifically tested that in the lateral habenula.
Do they use different types?
They did. They used paroxetine, that's a common SSRI, a selective serotonin reuptake inhibitor, and also ketamine, which is known for working much faster.
And what happened?
Remarkably, both drugs, despite having different primary mechanisms, managed to restore autophagy function in the lateral habenula of those chronically stressed mice.
Restore it. So bring it back up towards normal levels.
Exactly. The markers they measured, like seeing less of a protein called P62, which autophagy normally removes, and more of another called Beclin-1 involved in starting autophagy. It all pointed towards autophagy getting back online after treatment. This fits with some health news suggesting different antidepressants might converge on common pathways.
And did restoring autophagy affect the brain region's activity itself? I remember the lateral habenula is often overactive in depression models.
Yes, absolutely crucial point. They found that restoring autophagy coincided with a quieting of that neuronal hyperactivity in the lateral habenula,
Wow, so silencing that overactivity.
which could be a key part of how these drugs alleviate the depression-like symptoms.
So it suggests maybe the effectiveness of these different drugs partly hinges on their ability to fix this broken cleanup process in the lateral habenula.
That's certainly the strong implication. It points towards lateral habenula autophagy as a potential common node, a shared target for different antidepressant actions.
Okay, the study went even deeper, into the molecular nuts and bolts. These names came up: AMPK and mTOR, adenosine monophosphate activated protein kinase and mammalian target of rapamycin.
Ah, yes, the master regulators. AMPK, that's adenosine monophosphate activated protein kinase, and mTOR, mammalian target of rapamycin. Think of them as key control switches for autophagy inside the cell.
How so?
Well, AMPK generally gets activated when the cell senses low energy. It's like an alarm saying, okay, time to clean up and recycle resources. So AMPK tends to initiate autophagy.
Got it, the on switch.
Right. And mTOR, mammalian target of rapamycin, is more active when there are plenty of nutrients and growth signals. It essentially tells the cell, things are good, no need for major recycling right now. So mTOR tends to inhibit autophagy.
The off switch. Okay, so how did stress affect these switches in the lateral habenula?
Just as you might predict from the autophagy results, acute stress led to more active AMPK flipping the on switch, but chronic stress led to more active mTOR hitting the off switch.
Ah, so chronic stress leads to too much off signal from mTOR, suppressing the cleanup.
Exactly, and potentially not enough on signal from AMPK. It throws the balance off.
Did they test if these molecules were really causing the effects?
They did. For instance, they used a drug to specifically block AMPK during acute stress.
And what happened when they blocked the on switch?
Well, just as expected, it prevented the boost in autophagy usually seen with acute stress. But here's the really interesting part. Blocking AMPK also seemed to interfere with the brain's natural recovery process at the synapses, the connections between neurons, after the stress was over.
Wow. So that initial AMPK-driven autophagy burst might be crucial for resilience, for bouncing back properly.
It certainly suggests that. It might be important for preventing longer-term negative changes.
Okay, what about the off switch, mTOR? Since chronic stress activates it, did they try blocking it?
Yes, using rapamycin, a drug known to inhibit mTOR. And when they gave rapamycin to the chronically stressed mice, they saw antidepressant-like effects. The mice seemed less depressed.
And did it affect autophagy?
It did. It enhanced autophagy in the lateral habenula, just as you'd expect if you inhibit the inhibitor.
Makes sense. But did they prove that the autophagy itself was necessary for rapamycin to work?
This was a key experiment. They used genetic tools to specifically mess up the autophagy machinery only in the lateral habenula, reducing levels of a crucial autophagy protein called ADG7.
Autophagy-related gene 7.
Okay. And when they did that, guess what? Rapamycin's antidepressant effects basically disappeared.
Gone. Wow. So blocking autophagy in that specific spot prevented the drug from working.
Completely. It really nails the link. And it wasn't just rapamycin. They found something similar when they impaired lateral habenula autophagy. The benefits of both paroxetine and ketamine were also reduced.
So it really solidifies this idea that functional autophagy in the lateral habenula is critical for how these different antidepressants seem to work.
Precisely. It's a cornerstone of their beneficial effects in these models.
This all points quite strongly toward boosting autophagy there as a potential treatment strategy itself. Did they try that directly?
They did, and this is perhaps one of the most striking findings. They used a specific compound, a peptide called tatbeclin-1, which is known to directly activate autophagy.
Okay, so directly flipping the switch on.
Exactly. They infused this directly into the lateral habenula of the chronically stressed mice. And the results were, well, pretty dramatic. They saw rapid antidepressant-like effects within just 30 minutes.
30 minutes. That's incredibly fast compared to traditional antidepressants.
It is. And the effects were sustained. It suggests that directly targeting this cleanup process might offer a much faster route to relief, at least in these models.
And did it also normalize the neuron firing?
Yes, it helped quiet down that excessive firing in the lateral habenula that we talked about earlier.
Was it just a blanket effect or more targeted?
Interestingly, it seemed more targeted. The paper noted it was less effective at quieting neurons in non-stressed mice whose lateral habenula neurons weren't already overactive.
Ah, so like an on-demand system. It preferentially works on the neurons that are already hyper.
That's the suggestion, yes, which is quite an elegant potential mechanism fixing what's actually broken.
So what is autophagy cleaning up in these overactive neurons? The study mentioned glutamate receptors. Glutamate is the main excitatory signal in the brain, right?
Exactly. Glutamate excites neurons, and its receptors on the cell surface are like the docking stations for that signal. The researchers looked at these receptors on the lateral habenula neurons.
And what did they find under chronic stress?
They found an increase in the levels of key glutamate receptor subunits, specifically mentioning GluA1, GluA2, and GluN1 on the surface of these neurons. Basically, the neurons became studded with more docking stations, making them more sensitive to excitatory glutamate signals.
More sensitive, hence more active.
Okay.
And when they boosted autophagy with that T-AT-beclin-1 peptide?
They saw a reduction in those same glutamate receptor subunits on the surface.
So the enhanced autophagy was literally clearing out the excess receptors.
It seems so, like tidying up the docking stations to bring sensitivity back to normal. And they even showed that if they prevented the breakdown of these receptors, the antidepressant effects of boosting autophagy were blocked.
So those receptors are definitely key targets.
Yes, and they even use super high-resolution microscopy, stochastic optical reconstruction microscopy, to visualize the autophagy machinery actually interacting with these glutamate receptors right at the synapse. Direct physical evidence of the cleanup in action.
That's incredibly detailed. Okay, stepping back a bit, what are the bigger implications here? How does this change how we think about stress and depression?
Well, one major implication is reconceptualizing the stress response itself. The researchers proposed this fascinating idea that lateral habenula autophagy mirrors Hansel's classic general adaptation syndrome.
The alarm, resistance, exhaustion stages.
Exactly. The initial activation of autophagy by acute stress, that's the alarm phase. Then, the sustained effort by autophagy to remove excess stuff like glutamate receptors during ongoing stress, that's resistance.
And
Then, under chronic stress, when mTOR gets overactive and suppresses autophagy, that's the exhaustion phase. The system fails, neurons become hyperactive, and that potentially contributes to depression.
That's a really compelling framework. So therapeutically, this points towards new drug targets.
Absolutely. The most exciting potential is developing drugs that specifically enhance autophagy, maybe just in the lateral habenula. It could be a whole new class of antidepressants, perhaps faster acting and more sustained.
Could it also lead to more personalized treatments?
That's definitely a future possibility. Imagine if we could measure someone's lateral habenula autophagy function maybe through biomarkers or advanced imaging someday. You could potentially tailor treatment, maybe giving an autophagy-boosting drug specifically to those who need it.
And it might also explain why current drugs take time to work.
That's another very interesting angle. Perhaps the slower effect of traditional SSRIs is partly because they take time to gradually nudge this autophagy system back online in the lateral habenula.
Whereas ketamine might kickstart it much faster.
The study certainly hints at that possibility, linking ketamine's rapid effects to potentially quicker impacts on these autophagy pathways.
Now, we should also touch on the limitations, right? This is still early-stage research in some ways.
Absolutely essential to keep perspective. The biggest caveat is that this was primarily done in mice. Animal models are powerful tools, but findings always need careful validation in human studies. Brains are different.
And depression itself is complex, involving more than just the lateral habenula.
Right. While this study brilliantly highlights the lateral habenula, depression involves a whole network of brain regions. We still need to understand how autophagy functions or malfunctions in those other areas too. It's likely not the same everywhere.
Plus, autophagy itself isn't just one simple thing, is it?
No, it's a complex set of processes. This study focused mainly on one type, macroautophagy, in neurons. There are other forms, other roles that might also be relevant to stress and depression, so there's still a lot more detail to uncover.
Okay, so bringing it all together, what's your expert takeaway from this deep dive?
Well, synthesizing everything, this research provides really compelling evidence, I think, for a direct causal link between problems with autophagy in the lateral habenula and depression-like states, at least in these animal models.
Really?
The fact that existing antidepressants seem to fix this and that directly boosting autophagy works rapidly and robustly, that just opens up really exciting avenues for new treatments and further research.
Any particular finding that stands out?
For me, that on-demand nature, where autophagy seems to target the most hyperactive neurons, is particularly elegant and has broad implications for just maintaining brain health in general. And also, the idea of using AMPK or mTOR activity levels as potential biomarkers for stress stages or depression subtypes.
Yeah.
That could be huge down the line.
Yeah, definitely. This has been incredibly insightful. So for our listeners, what's a final thought to leave them with? Something provocative based on all this?
Maybe consider this. How might the chronic stressors in your own life be impacting these fundamental cellular housekeeping processes in your brain, like the autophagy we discussed? We often think about stress emotionally, behaviorally, but this shows a real, tangible biological impact. Could actively supporting these natural repair systems through lifestyle, stress management, maybe future therapies be a key part of building resilience, of protecting ourselves?
That's a really powerful question to mull over. If this has sparked your interest, definitely look into resources on stress management, or keep an eye on the ongoing research into autophagy and brain health and these novel antidepressant approaches.
It really underscores how complex and adaptable our brains are.
Absolutely. And how science continues to light up new paths towards understanding and improving well-being. Thanks for joining us on this deep dive.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.